Welcome to Shiny LipidSearch. So here we'll be looking at some MS spectra. The output from LipidSearch, and since we've only got one data set, I've made it available here so you can directly choose the file. You don't have to upload one. You can of course upload a different one. But here we're going to load one of these tab separated files. So this is the negative mode, the first one that contains a lot of data. So here you see everything. You can select more columns, extra columns, delete some columns you don't like, if you do, and so on. The next thing you can do is to run a linear model. And there are a few options here which I'll explain in a moment. But let's just see what happens if we use the default settings. So this is now for the lipid class ceramides. Run the model. And here we get all the points that are on the grid. So how do these points get selected? Initially there are a lot more points. The model gets kick-started by using always the smallest retention time if there's several for the same kind of lipid, which in this case would be the lipid with, say, 40 carbons and 3 double bonds. There might be several in the data set, but it will initially select the one with the lowest RT, and then after the first round it will select the ones that fit the model best, and it will carry on until the model doesn't change anymore. In this way, sort of for each lipid there's only one representative. You can adjust several things here. One is sort of the minimum or the maximum number of double bonds, the minimum or the maximum of the number of carbons. You may not have seen this before. You can both change the lower and the upper range. Yeah, maybe something I haven't used before. And you can use, this is the tolerance for sort of which points that don't fit the grid very well, how far they're allowed to be off. So in this case, they're allowed to be off by 0.5 minutes. So these things, usually you don't really need to change anything. You can use this to get rid of some points. Also these that maybe some don't fit so well. What's more important really is what's down here. So say for the same ceramide 40.3, there might be not only the main ion, but a second ion. Ion here means adduct. So the main adduct for this group is acetate. But there is, for some of them, there are also the H- ones. This is negative mode. So if we want those as well, we say include ion in the model. That means these will now, sort of for every possible ion, there will be one representative in the model, if there is one. So let's see what happens. Do we see any extra ones? There are a few extra ones. And so in this case, they fit very well. We can click on them and see whether we see what they are. So here we see it's the, so where does it say ion? Yeah, it's the acetate ion. This one then is the minus H ion. There we go. And in addition, you can also include decoration. That means that will also increase the number of points in the model. So ceramides have this decoration either D or T for di and tri. I think hydroxylation. I'm not entirely sure what it is. So di. So the D is the dominant species. If you don't include it in the model, then only the dominant species for both ion and decoration will be chosen. And so if you run this now, you should get some extra ones. And there we are, and we may have seen. So this is D, and we should see a T as well somewhere. No, we don't see a T. Let's see. We had a new feature here. If you draw a box around something, it will zoom. And hopefully we can now identify what we're looking for. So this is a T. Indeed, here's a T. We found a T. So there's T, 42.2T, 42 to D, and 42 to D M minus H. These are all the 3 points. If you want to get back to the normal view, you double click. And so the last thing that you can do is you can say, you can include a grouping factor for fatty acid. So fatty acid is not fitted in the model because there are too many, but you can use it to, you can still, if this 42D has a different composition than, say, this 18.124.1, say 18.2 and 24.0, then you will get a second point. If both of them exist and you press this one, group by fatty acid, then you should get 2. Whereas if you don't do it, you only get 1, you get the best one. So let's do this and see what happens. So we get some extra points. And if we now zoomed in, we would probably find that there's several species of this. And this you can do for all the different lipid classes here. Remember to click Run Model, otherwise nothing changes. You can change the size of the thing, blah, blah. You can change the size of the thing. You can download the plot. You can download the data. In the data, if you download the data, it will download an Excel sheet with Excel file with 2 sheets. One is the data as it is that is used, so that has been filtered for the criteria for sort of the best fit to the model according to these criteria. But there's also a second sheet called plot data where the contributions of decoration and adduct have been calculated out. So in this model here, in this plot, for example, the decoration D and T appear quite close together, but their retention time might really be quite far apart. But if the difference on average between the D and T species is, say, 0.5 minutes, then this difference will have been subtracted in plot data so that they can be plotted in the same plot. Otherwise, they would be too far apart. So so the 2 sheets are not exactly the same. In plot data, the effects of the 2 factors, iron and decoration, have been removed by subtracting them. So I hope that I've covered everything, and yeah, I hope you can explore the data with this.